So we are happy to have with us today Dr. Davinia Perdomo-González and Dr. Nora Garcia. Uh, we're going to have two presentations today in the time that we have. And we're going to start with Dr. Davinia. Dr. Perdomo-González received her bachelor degree in veterinary medicine from the University of Las Palmas de Gran Canaria, Spain, and has recently finished her PhD from Sevilla University in quantitative genetics. She is currently a researcher in the Miragem Research Group and is responsible for the annual genetic evaluations for the Pura Raza Española and Pura Raza Menorquina horses, but also collaborates in the genetic management of other autochthonous equine breeds of Spain. She has two main research focus areas. First, the analysis of populations' genetic structures based on pedigree information, and second, the implementation in equine breeding programs of new methodologies developed to estimate non-additive genetic variants, such as parent of orange effect, dominance deviations, or inbreeding depression loads. Looking ahead, her professional aspirations align with her continued commitment to animal research and genetic improvement. Thank you very much, Davinia, for being here today with us, and the floor is yours.
Thank you very much, Sanla, for that amazing presentation. Let me share. Do you see my screen?
Yes, perfect.
Okay. Yeah, well, that's it. I am Davinia Perdomo. I come from the University of Seville in Spain, and I'm going to introduce you in the genetic analysis of the genetic analysis of the population structure and inbreeding in the Pura Raza Española horse from a quantitative point of view. The Pura Raza Española horse was established in 1912 with the creation of the breed studbook. From that moment on, the army played a significant role in its management. Then, in 1972, the Royal National Association of Purebred Spanish Horse Breeders was established. From this point on, there was a transformation in the use of the horse. Previously associated with the military, agriculture, and transport work, it began to be used more for leisure and sports. In fact, in 1996, the Pura Raza Española horses started to compete in the Olympics in classic dressage. A few years later, a royal decree was published which completely changed the equine sector in Spain, providing breeders' associations more control over breed management, and a lot of specific regulations were created, which established the basic requirement for selection schemes and performance tests for the genetic evaluation of purebred equines. Then, in 2003, the first agreement was made with Miragem Research Group, which is responsible for the genetic management of this breed. The Pura Raza Española horse is the most widespread breed in Spain, distributed throughout all its autonomous communities, and is also present in more than 65 countries. As you can see, Andalusia is also, where the Pura Raza Española horse originated, has the largest census, about 30%, while another almost 30% is abroad, mainly in the United States and Mexico. Nevertheless, the Royal National Association of Purebred Spanish Horse Breeders is the worldwide association and the only one officially recognized to guarantee purity through the management of its studbook and breeding program. The breeding program includes both the breed regulation and the improvement program. The three main objectives of the Pura Raza Española Horse Improvement Program are the preservation of the breed standard, both from a morphological and functional perspective. Secondly, the production of healthy animals without hereditary defects, which is crucial in a closed breed like this, where there can be significant problems of loss of genetic variability and inbreeding. And finally, the improvement of morphology and functionality focuses on equestrian sport, mainly in classical dressage. The total database of the Pura Raza Española horse population was composed by more than 328,000 animals born from the beginning of 1900 to 2018. The pedigree competence level, so the proportion of ancestors known in age ascending generation, of the Pura Raza Española horse born in the last 10 years, the last generational interval, is over 99%, remaining about 90% until the 7th generation interval. Then, when we analyze the mean complete, mean maximum, and mean complete equivalent generation, we realize that animals born in the last generational interval have on average one more generation known than the total population average. As you can see, for some animals in the last generational interval, we know ancestors for more than 18 different generations. Then we examine the variability parameters. We can see that the average inbreeding and average relativeness coefficient were similar in both cases, an average inbreeding of 7.5% and average relativeness of 11%. These values were lower than those reported by other authors analyzing the same breed. This is due to the fact that this author did not include data from the last 2 decades, the last 20 years, in which more than 70% of the Pura Raza Española horse has been born, and in which the management by breeders and the breeding program have become a key part of conservation genetics aimed at avoiding mating with high levels of inbreeding. In this figure, you can see the Pura Raza Española horse population evolution by generational intervals in blue bars, highlighting a major increase in horses born over the last 20 years, in which more than 200,000 horses were born. It can also be seen the average inbreeding evolution over the time in solid green line, with a maximum value of 8.4%, and then it decreases to the current value of 7.25%. Also, the average relativeness in solid orange line behaved in a similar way, with each generation increasing and a minor decrease over the last 2 generations, around to 11%. As I said before, since the stud book creation, the registration of new Pura Raza Española horses has been restricted to those born from parents already registered. That's it. It has become a closed breed where the mating of related animals is inevitable, and therefore there is an increase in inbreeding. This is not only implies a greater resemblance between individuals, or in other words, the loss of genetic variability, but can also amass recessive deleterious alleles. As you probably know, the inbreeding of an individual is defined as the probability that an individual possesses in a particular gene 2 identical copies derived from the same common ancestor. This calculation has historically been made as the sum of the partial inbreeding of the family founders, but this doesn't provide detailed information about the contribution of each ancestor to the probability of identity by descent of the individual. To overcome this, a methodology was developed whereby the inbreeding of an individual can be broken down as the sum of age of the partial inbreeding coefficient it receives from age of its common ancestor, and it can be described as the combined probability that an individual I is autozygous for an allele, and that allele was derived from the ancestor J. This calculation was made possible by the methodology developed by Casellas in 2018 and the program implemented by Varona and collaborators in 2019. This new approach made it possible to break down the inbreeding load in an individual into the sources of ancestry among its parents, including the founders of the population, the traditional approach, and Mendelian sampling of the non-founders, a new approach. The partial inbreeding coefficient estimation resulted in a database with more than 58 million records, one record for each partial inbreeding coefficient that each animal receives from a given common ancestor. And as you can see at the same figure than before, the average partial inbreeding coefficient that a common ancestor transmitted to their descendants has seen a gradual decrease over the generation, the black solid line, to almost zero. But the number of common ancestors that transmit inbreeding to its descendants in yellow bars has increased, as expected, in fact. The fact the average partial inbreeding coefficient that common ancestors transmit to its descendants have decreased is mainly due to the increasing awareness about among horse breeders about the inbreeding depression and the better information available about the animal's breeding values. Enables them to choose the best stallion and mares, also with the lowest average relativeness, to obtain foals with the desired characteristic and less inbreeding, thus reducing the average founder's contribution to the population. Nowadays, despite the large Pura Raza Española horse census, just 1,023 ancestors account for 100% of the genetic variability. And the 50% of this genetic variability can be explained by only 7 ancestors. These 7 most influential individuals transmit an average partial inbreeding coefficient that ranges between 0.03% and 0.74%. And despite of having a large number of descendants, to whom they transmit inbreeding, very few of them have received a partial inbreeding coefficient equal to or higher than 6.25%. Once the population structure was analyzed, the need to obtain reliable inbreeding values for better population management became even more evident. For this purpose, a comparison of different methodologies for the estimation of inbreeding was made. On the one hand, those based on pedigree, and on the other hand, those based on genomic information. Genealogical inbreeding coefficients are based on pedigree data, and therefore depend on the depth and completeness of the pedigree, generating estimated values. Nevertheless, by the time genotyping costs have come down, genomic inbreeding coefficients can be easily obtained, which are expected to be true measurements of an individual's level of homozygosity. At the time of this analysis, the number of animals in the pedigree was over 340,000 individuals, of which at least 3 complete generations were known for over 97% of the individuals. From a genealogical standpoint, classic bright inbreeding, Balow's ancestral inbreeding, Kalinowski ancestral inbreeding, recent Kalinowski inbreeding, and the coefficient of ancestral history were analyzed. Also, depending on the number of generations taken into account, classical inbreeding coefficient at the third, sixth, and ninth generation were also computed. Furthermore, based on the genomic information of 805 animals, individual inbreeding coefficients were determined, and coefficients were also estimated using the method of moments, the diagonal element of the genomic matrix, and the diagonal element of the hybrid matrix. When we analyze the evolution of the different pedigree-based inbreeding coefficients through history, they all appear relatively constant until around the year 1940, when they start to increase. The mean values ranged between 0.01 for classical inbreeding at third generation and 0.08 for the bright classical inbreeding. On the other hand, the historical ancestral coefficient and Balow ancestral inbreeding have grown exponentially to reach values about 0.3 and 0.5. The genomic based inbreeding average values show a clear difference based on the methodology employed. Within the row-based methodology, the average value oscillated between 0.05 to 0.17. In comparison, the matricial and method of moment-based methods showed lower and very similar values, all of them close to 0.05. Pearson correlations between genomics and pedigree-based inbreeding coefficient were mostly significant, positive, and large in magnitude, ranging from 0.58 to 0.79. The classical pedigree-based coefficient showed a good fit with the non-matrix genomic values, while the matrix genomics coefficient showed significant correlation with pedigree data only when recent inbreeding is taken into account. Finally, both Kalinowski coefficients showed mostly high and significant correlation with the rest of genomic values. In conclusion, I think that the take-home messages are the Pura Raza Española horse has a deep and reliable pedigree studbook. The Pura Raza Española horse breeders have increased their awareness about their population inbreeding, and they are working on reducing their average relativeness. And even when real parameters are preferable, classical pedigree parameters remain useful for reproductive management in population with deep and reliable pedigree. Thank you very much.
Thank you, Davinia, for your presentation. We're gonna hold the questions, everyone, for the end of both presentations. And now we're gonna have Dr. Nora presenting for us. You can put your slides while I read your bio. Is that okay? So, Dr. Garcia holds bachelor degrees in biotechnology from the University of the Basque Country. She completed her PhD in equine genomics at the University of Cordoba in September of 2023. Currently, she's a researcher in the Merahen Research Group and collaborates with the Genome Task Force led by the Royal National Association of Purebred Spanish Horse Breeders in design an equine genotyping chip. Her main areas of research include inbreeding and genomic homozygosity, whole genome association analysis, distortion of allelic transmission ratios, analysis of genome copy number variations, and the search for candidate genes related to numerous economically important traits in horses for subsequent implementation in equine breeding programs. Her future perspective involves continuing genomic animal research. Thank you for being here with us today, and the floor is yours.
Nora, el micrófono.
Thank you for the presentation, Sanla.
Oh, you're welcome.
Okay. I'm Nora from University of Córdoba, and I'm going to talk about genomic inbreeding and inbreeding depression in reproductive traits in mares. As Davinia explained, the Pura Raza Española horse has a closed breeding system in which only animals belong to the breed can act as breeders. Therefore, the mating of related individuals is difficult to avoid, generating a loss of genetic variability, which can lead to an increase of inbreeding in their offspring, leading to the appearance of detrimental phenotypic effect. One of the main causes of poor reproductive performance in horse is the increase of inbreeding in population. This genetic phenomenon, associated with the loss of aptitude and average phenotypic performance due to mating between relatives, is inbreeding depression. Traditionally, the study of the effect of inbreeding in animal population was carried out with genealogical data. However, nowadays, the increase in the number of genotyped animals allowed to carry out the study using genomic estimates based on random homozygosity. Their analysis allow us to study genomic homozygosity and to associate a higher homozygosity in a specific genomic region with a phenotypic effect. In the first study, the aim was to analyze the relationship between molecular homozygosity and fertility in Pura Raza Española mares, and determine the existence and putative function of genomic region where there is increased homozygosity that could affect fertility. The initial reproductive dataset includes 340,707 foaling, a mare whose main activity was not breeding, as well as mare primarily engaged in leisure or sporting activity, were excluded. As a phenotypic trait, we used the reproductive efficiency recently estimated by Perdomo-González et al. And defined as the percentage deviation between the number of optimal and actual foaling of the mare at each age during her productive life. The estimation of genetic parameters was performed using a real animal model using the program BLAB F90, and the pseudo-phenotypes were estimated using the methodology proposed by Garrett et al. 890 horses were genotyped with the Action Equine SNP genotype array high array density. Genotypes were analyzed and filtered with Action Analyze Suite software and Plin. The core rate used was 0.95. No map or LinkedIn disequilibrium filtering was used as recommended for row analysis. The final genomic dataset was 540,294 SNPs on 31 chromosomes. To estimate the genomic inbreeding coefficient, we determined the rows per animal using the slightly preserved with the datagram package. And finally, genomic inbreeding per individual was estimated at the genome and chromosomal level. In addition, genomic regions in which the incidence of RO was significantly increased were determined by a permutation test. This region greater than 100,000 base pair that saw a 100-fold increase compared to a random probability of occurrence. These regions were validated in the whole population, and finally, using a t-test, the association between each row region and efficiency reproductive values was determined. The region with T greater than 2 were significantly associated with male fertility. As for the functional analysis, genes of significant regions were annotated with BioMar, a reference genome, and functional annotation was performed with David and Pater. The pseudophenotype saw a heritability of 0.25. The pseudophenotype values of the genotype individuals saw a normal distribution and a wide range of variability. This divergence allows us to perform homozygosity analysis at the total population level and dividing the population into high fertility and low fertility animals. The average of genomic inbreeding in the whole population was high, 0.136. However, it was almost 30% lower in high fertility animals compared to low fertility animals. Regarding the analysis by chromosome, the results were similar. High fertility animals saw lower fraud than low fertility animals on 29 chromosomes. Regarding the correlation between reproductive efficiency and genomic inbreeding, there was a negative correlation with moderate but highly significant magnitude. The correlation between fertility and homozygosity at the chromosomal level were highly variable. Moreover, the highest correlation between reproductive efficiency and genomic homozygosity were obtained in whole genome analysis and not by chromosome. This suggests that in polygenic traits such as related to fertility, homozygosity values should be taken into account globally as predictor of inbreeding depression and not at the chromosomal level. A chromosome with the strongest correlation was chromosome 15, and the weakest correlation chromosome 18 and 29. And the correlation between reproductive efficiency and genomic inbreeding coefficient were only significant in 18 chromosomes of the total population and in 15 of the reduced population, suggesting the existence of genomic regions more or less involved in the genetic control or fertility. 41 genomic regions on 70 chromosomes were detected in the reduced population that saw a homozygosity rate more than 100 times higher than the casual occurrence rate. 13 of them were validated in the whole population to be associated with change in fertility, located in chromosome 1, chromosome 3, chromosome 4, chromosome 7, chromosome 20, and chromosome 30. Finally, 70 genes previously associated with biological process related to female fertility, including oocyte and embryoloid development, were annotated in 3 of these regions, located in chromosome 3, chromosome 7, and chromosome 25. After demonstrating that there was a negative correlation between increased genomic inbreeding and fertility in Pura Raza Española mares, the second part aimed to evaluate the intensity of inbreeding depression in 5 reproductive traits, using pedigree-based inbreeding coefficient and inbreeding coefficient based on RO. For this study, we started from the same initial data set as the previous research and performed the same exclusion of mare. The final data set was 29,847 animals, and the phenotypes were depurated and corrected, obtained predict residual phenotypes by REMEL methodology using phenotypic methodological data and SGIL methodology using genomic data. The 5 reproductive traits studied were age at the first falling, average interval between fallings, total number of fallings, productive life, and reproductive efficiency. In addition, 1,018 animals genotyped with neogen medium density chip were used. The classical inbreeding coefficient was estimated with the ENDOC program and genomic coefficient with the Telegram in R. To evaluate inbreeding depression, a regression estimation of predict residual phenotype of the trait of interest was performed. 2 methodological approaches were used. The first was a classical linear regression with SAS pressure, and the second was a segment regression with the nil pressure. Pedigree-based inbreeding coefficient and row-based genomic coefficient were used as regressor variable. Finally, the two breakpoint test based on autoregressive analysis with the autoreg procedure was used to statistically evaluate whether the structural change in the relation between the dependent and independent variable. The correlation between pedigree-based inbreeding coefficient and row-based genomic coefficient was moderately high compared to other breeds. This is due to the fact that our breed has more complete and reliable and deeper pedigree record compared to other horse breeds. Our results show that using genomic data in the model improved the estimate of the predict phenotypes. We saw that there is a high significant inbreeding depression effect of most reproductive traits, except for the average interval falling trait when pedigree-based inbreeding coefficient was used. And we observed that genomic inbreeding coefficient explains inbreeding depression better than pedigree-based inbreeding coefficient. Finally, we observed that the age at the first falling and average interval falling increase with increase in breeding. However, the number of falling is lower, productive life is shorter, and reproductive efficiency decreases as inbreeding increase. This result in lower reproductive aptitude and worse reproductive performance. Finally, a segment regression analysis was performed to evaluate the effect of inbreeding depression along the genealogical and genomic inbreeding range with the predictive phenotype with genomic. These results show that the level of genealogical and genomic homozygosity doesn't manifest homogeneously its effect on reproductive trait. That is, variation in the level of inbreeding depression were observed as the level of homozygosity increased, demonstrating that the reproductive parameters are sensitive to inbreeding depression even when homozygosity levels are relatively low. To conclude, a negative relation has been demonstrated between increased genomic homozygosity and fertility in Pura Raza Española mares, with the existence of genomic region where increased homozygosity significantly affects mare fertility being detected. Within this region, several genes related to reproductive process in various species have been identified. The use of genomic information in RML model to predict residual phenotypes improved the estimation of the influence of inbreeding on the static reproductive trait, compared to models that don't include genomic in their framework. A genomic inbreeding coefficient captures more inbreeding depression than pedigree-based inbreeding coefficient, although in the case of reliable pedigrees with high depth, both types of coefficient yield similar magnitude for this parameter. Our study has demonstrated the existence of inbreeding depression in all 5 studied reproductive traits, negatively affected by increasing inbreeding, both at the genomical level and through genealogical inbreeding. Similarly, it was observed that the reproductive traits are highly sensitive to inbreeding depression, with a negative effect on the phenotype of such trait, even at relatively low levels of homozygosity. Finally, the use of advanced statistical techniques such as segment regression has allowed us to determine the evolution of inbreeding depression effect across the range of analyzed level of homozygosity. Our study has shown that the increase in genomic inbreeding as well as genealogical inbreeding doesn't uniformly affect reproductive traits, exhibiting a non-linear effect as the level of homozygosity increase. And thank you for your attention.
Thank you, Nora, for your presentation. It's very informative. And now if anyone has questions, you can raise your virtual hands and turn on your cameras and microphones. Or you can also type your question in the chat and we can read them out loud. So Davinia, I have a question. I was curious. You showed that when you estimate the inbreeding using different techniques with the genomic data, you had like you had different results. And I was wondering why, like, do you have an explanation why? Because the difference was pretty high. It was not like small differences. And I was wondering if you have an explanation for that.
You are asking about the difference between some pedigree-based parameters and two of them that were higher than the others.
Yeah.
This was the definition, because those parameters are not really, well, one of them, the ancestral value coefficient, take account both the recent inbreeding and the ancient inbreeding. So it no measure the probability. And also the ancient history coefficient is a number that tells you how many times some allele has been inbreeding by descent over the generation. So you cannot compare the magnitude of them because they are measurement different things.
Okay, I got it. Nice. So we have Flavio. Go ahead.
Hi, Nora and Davinia.
Hi.
This is a comment and a question for both of you. So I note in your presentation, by the way, great presentations. Thank you for doing that.
Thank you.
I noticed that you use very kind of loosely homozygosity and inbreeding as the same thing, right? And in fact, homozygosity is not the same thing as inbreeding, because inbreeding, the concept of inbreeding is identical by descent, and not all, let's say, markers that are homozygous, their alleles are identical by descent. And this is very important when you compare pedigree-based inbreeding and genomic-based inbreeding, because all measurements of genomic inbreeding are based on homozygosity. What you are measuring is homozygosity, how homozygous is individual or between two individuals, the homozygous given region, right? But when you use the expectation based on the pedigree, that expectation represents what we expect to be identical by descent. The alleles transmitted exactly from the same ancestor. So in this sense, you expect different magnitudes of inbreeding, right? So because one is the genome is based on homozygosity, it should be higher. The expectation should be higher than based on identical by descent. So I think you have to really think about this and when you talk about inbreeding, when you use the genomic. And that's why the results are quite different, right? And it's not a surprise to me that when you look at inbreed depression, that you use a measure of homozygosity, that total homozygosity using the genomic, that you're going to have a stronger association with depression, right? Because you are capturing more of the homozygosity. But the concept in breeding is important because what changes, the change that you see over time in a population that increases the homozygosity, that increases due to identical by descent. It's not identical by state or chance. So I think when you compare methods, these two methods, pedigree-based and genomic-based, what you have to look at is the difference across generations, across years. That will be much more consistent, regardless the method that you use, right? Because the changes are expected to be due to what is identical by descent. Do you understand what I'm saying?
Yeah. I totally agree with you. But for example, in some methodology, for example, the Van Reyden formula take account the identity by state probability of homozygosity. So some of methodologies take account that not all the homozygosity is inbreeding by descendant.
Based on genomic relationship metrics, the genomic relationship matrix is just sharing of the same alleles, not identical. It's just homozygosity. And Van Reyden is based on just the genomic relationship that is just based on homozygosity, the sharing of the same alleles, but it doesn't mean that it's sharing from the same ancestor. Two, right?
It could be.
Yeah. That's why the scale of the genome relationship is not exactly the same as the identical by descent. It's not just because of the observed segregations, because the scale is different. But yeah, no, it's just a comment. It's not that great work. I really like it to see the results. It's just you have to be careful because like runs of homozygosity depend on if you are looking at your criteria to look at long or short runs of homozygosity. If you exclude, for instance, short, you are going to be just look at the very recent homozygosity, therefore recent inbreeding, right? If you look at the small runs of homozygosity, you look more to the past or ancient. And all these will have implications on the interpretation of the results and how similar will be the pedigree-based and the genomic-based inbreeding that you calculate. Just from some food from thought.
Yeah.
First to think about. This is something that I am always thinking about. And when the students, they come and say, well, I get very different inbreeding coefficients using one or another, say, yeah, but that's expected. So why you don't calculate the changes over time? Then when you look at the change over time, they're quite similar. The rate of change.
They change over time. I don't understand your question.
No, the change over time. Like you don't look at the inbreeding coefficient itself. You look at how the inbreeding of population is changing over generations.
Yeah.
Right? So when you do this based on genomic information or pedigree information, usually these changes are very similar, much more similar than the coefficient themselves. The inbreeding coefficient based on genome will be higher. But that's expected because it's homozygosity, the total homozygosity, right?
Also, it could be because the animal that we can genotype are recent animal because we need a sample of them. We cannot measure the inbreeding of a very old
but
animal.
Even if you have an animal with the same, you don't have this problem, it's still the inbreeding coefficient based on genomic information will be expected to be higher. the magnitude. But if you look at the changes over generations, is a much similar amount, because the changes are due to identical by descent, right? In both cases, if it's pedigree based or genomic based.
Okay.
Okay.
Okay. Well, I
Thank you.
have to give a chance to others to ask questions. Thank you so much, Nora and Davinia, for doing this. Good work.
Thank
Thank you
you.
for the presentation.
Thank you.
Thank you, Flavio, for your questions. Does anyone else has questions for Davinia or Nora, or Davinia and Nora? Feel free. I was thinking here, like, I know how I don't know, but I imagine how horse breeders, they can be very closed, like on taking new information, just like any other breeder or farmer. I was wondering if you guys shared these results with them about how inbreeding can impact reproduction, especially, and if you did, how was the reception? Like, how was like the dissemination of information to the breeders?
Yeah, the Meragen Research Group publish each year the genetic evaluation of the breeders, and so the breeders can search the animal. Also, they are published in a website where each farmer, I don't know how to say it. Each breeder search their animals. They can see all the genetic breeding values. Also, they can check for possible meetings. They can search for a stallion and a mare and look what the inbreeding of the future foal could be.
Okay.
There is a tool in the website and where they can see all that information.
Oh, this is
Breeding
nice.
about morphology, about functionality, inbreeding, also sport results.
Okay, nice. Yeah, perfect. Okay, so last call for questions. If we don't have any more questions, we're gonna let Nora and Davinia.
I have one more. What is going to. So,
Okay, of course.
Yeah, I just wonder about this breed. Is this breed for what is the purpose? Is racing?
Morphology for functionality in mainly for classical dressage.
Classical? What do you mean?
Like a dance.
Oh, dance. Oh, okay. So for presentation, like dance. Oh, okay.
Like eventing.
Okay. I was not sure. I never heard about the breed before, but I look at the pictures, so that it was really beautiful animals, right? For dancing. Mm-hmm. Okay.
Yeah.
So so for this, why I asked this because I worked with racing horses before, like some in the past, and because of for horses that race, the explosion is very important, right? The energy, like that explosion of the muscles and to run very quickly in a short time. So the mitochondria in the cell is a very important. The number of mitochondria in the cell, right, to produce that energy that you need. So that's why empirically the producers always give a lot of attention to the mare, sometimes more than the stallion when selecting. And that's because the mother transmits the cytoplasm, right? So that's why I would, I thought if it was a racing horse, I would ask you if you observe this in the breed, like more selection that based on the mare than really the stallion. But in this case, I think for dancing, you don't need that level of energy, the explosion. But it's very, very interesting. That's what something that see why the industry is so focused on the mares, right? In selection these families and based on, but then now I really understand why this is. So it's science behind. It's really to identify these mares that really transmit this cytoplasm that is rich in mitochondria. So that would that is the powerhouse of the cell, right, in terms of energy for this short burst of energy that's needed to run, right, very quickly in a few 100 meters or 2,000 meters. Okay, so
That's
thanks
interesting.
Again.
Yeah.
Thanks again.
Thank you again, Flavia, for your question. Okay, so we would like to thank you girls again for doing this to us. It's very nice to have former like visiting students presenting for us, and we'll be more than glad to have you two in the future again.
Thank you very much.
Thank you. Thank you.
Okay, bye.
Bye.